Everyone, could you see my first slide?
Yeah, it's all perfect.
Okay, that's great. So my name is Olivier Duclos, and I work at Sanofi. I'm very pleased to be with you today, even it's in a remote mode. Hopefully we will be able to meet in the near future. And firstly, I'd like to thank the organizing committee for giving me the opportunity to share with you a kind of overview of the challenges and advances in antibody-drug conjugates, ADC, namely, which is, as you probably know, a growing field in the pharma industry. And hopefully we'll keep delivering some new efficient treatment for patients. So please let me remind you what we are talking about. What is an ADC? It is actually composed of 3 components: an antibody that targets a tumor antigen that ideally should be specific of the tumor. There is the highly potent cytotoxic drug that needs to be delivered inside the cell, the tumor cell, obviously to kill it. And then a linker that allow actually to connect the drug to the antibody that will be cleaved, obviously to release the drug within the cell. So as you also likely know, the concept of ADC is really to increase the therapeutic index of cytotoxic drug that actually could not be used, as it was proven in clinic in chemotherapy due to their high toxicity. And as depicted in this graph, ADC format actually allow to both increase the maximum tolerated dose through the selective delivery of the cytotoxic to the tumor cells, and also to decrease what is called the minimal effective dose, the in vivo efficacy, if you will, through optimal delivery of the cytotoxic into tumor cells, obviously for killing them. And at this stage, I think it's very important to raise 2 important challenges that we will see later on. The first one is that we are really far from the so-called magic bullet behavior, since you can see that there is a very limited amount of the injected dose of ADC that is able to reach the tumor. And the other thing is that the tumor retention is also limited due to the nature of the antibody scaffold. So actually, in the last decade, ADC modality has become really a therapeutic success in oncology, with the approval not of 7 but of 8, since there is a last approval that came up in April, which are actually Adcetris in 2011, Kadcyla 2013, Besponsa and Milotag in 2017, and actually 2019 has been a great year with approval of Polivy, Enhertu, and Padcev. And maybe I just want to highlight the fact that the Milotag drug was actually initially launched in 2000. It was the first one actually, but was withdrawn and reapproved following actually a different schedule through dose fractionation. And as I said previously, in late April, as you can see, the FDA approved Trodelvy from Immunomedics for the treatment of the triple-negative breast cancer. And I think it's very interesting and promising for patients to note that there is an increase of ADC for treatment of solid tumors, which is actually considered as being more challenging to tackle than, liquid. And mechanism of action of cytotoxic drugs that were approved by the FDA. It comprises, as you can see, mainly DNA and tubulin binders, and more recently we appeared to topoisomerase inhibitors that we will come back later on. On the next slide, I would like to share with you a kind of a brief overview on what could be the next approval across the most advanced ADC. As I mentioned previously, Trodelvy should not be not anymore in this slide, but I did not have the time to redo it. But we do have, following this nice approval, still 7 remaining ADC at advanced stage of biologic license application, phase 3 or pivotal phase 2. And most of the payload of those, advanced ADC, actually, as you can see, tubulin binders in green and DNA binders in red. And once again, I think it's important to stress that there is really an increase of ADC developed for the treatment of solid tumor, because it was really something that was not given, very honestly, at the very beginning. And there is still a lot to do in that field. The next slide, sorry, shows you that, whoops, sorry, this one. Sorry, maybe, yeah, okay. So the next slide shows you that, as I said, ADC field is active and growing with several successes that we already talked about, but it is, I think, to be stressed, to be fair, a high rate of clinical failure. And this graph actually displays the number of new active, discontinued, and approved ADC in the clinics since 1977 until 2019. The results with the approval of Trodelvy in 2020 is not yet actually included. As you can see, a total of about 160 ADCs have progressed into the clinic since 97, and 76, that means about a rate of failure of 38%, have been discontinued. And something that is not visible on this graph is that most of the ADCs that were discontinued occurred actually in phase 1. So actually, in order to improve the efficacy of ADC, it's important to understand the different steps of the mechanism of action of ADCs that leads to the tumor cell killing. And the first step is really the binding to the antigen present at the cell membrane, followed by an internalization of the complex through endocytosis. And during the cytosolic trafficking, the formed endosomes evolve in acidic lysosome, where the processing of the ADC takes place. The drug is released into the cytosol to kill the cell, but it could also be released outside the cell to kill a neighboring cell, and this is called the well-known bystander effect, which is actually a double-edged sword, since it could increase efficacy by killing, additional tumor cells, but it could also cause toxicity by killing actually neighboring normal cells. And importantly, last feature which is important is the fact that the antigen could be recycled at the surface of the cell and be re-engaged in the whole process. For efficacy, what we know is that antigen density, internalization, kinetics, and recyclings are really key parameters. And for example, we know that lower levels of antigen expression may be compensated by increased internalization and trafficking efficiency. So it's really an interplay between various parameters that should be taken into account. So optimization of an ADC is a very complex process that requires actually to play with many interdependent parameters, and this is, I think, well reflected by the high rate of clinical failure in the field. And as shown on the slide, key parameters should be considered for, various key parameters should be considered actually for each component of the ADC. For example, antibody should have a high specificity, optimal binding affinity and good PK properties. The linker obviously needs to be stable in circulation to avoid premature release of the cytotoxics, and cleavage needs to be efficient in the cell, but it could also sometimes in some design take place in the extracellular compartment. Cytotoxic drug needs to be highly potent with a good susceptibility of the tumor to its mechanism of action, and physical properties also a great impact on the TI, as well as the loading, the so-called drug antibody ratio named DAR. And overall, I would like to stress that the success in the design of new successful ADC relies really also on technology advances in multiple fields, and we will see some example later on. So selection of the payload is obviously of great importance. And as you can see on this slide, there is a broad range of mechanism of action being used to develop ADC, DNA binder, tubulin binder, RNA polymerase, and topoisomerase. So tubulin binders and DNA binders are really the most popular and I would say one of the most potent payloads. And there is also an emergence with the success of actually, as I mentioned previously, Trodelvy and HER2, there is an emergence of ADC based on, I would say, moderately potent payload. And it's really a change of paradigm since we thought previously that it was necessary to have very, very potent payload for achieving efficacy, but to the extent of the toxicity profile, of course. So in the selection of the cytotoxic drug, it is really critical to match the right mechanism of action with the right indication. And to illustrate this statement, we did some internal experiments showing that through a head-to-head comparison of a tubulin binder ADC versus a DNA binder ADC in a lung PDX model and a colon PDX model, we were able to show that in the lung PDX model, the tubulin binder ADC was almost inactive, while the DNA binder ADC is pretty active. And in the colon model, this is almost the opposite, since the tubulin binder is more active. And this is obviously associated to the level of expression of the antigen for each tumor. So that means this is really combinations that needs careful attention to be able to design a very efficient and safe ADC. The selection of the linker is also key for efficacy because it actually governs what is going to be released in the cell. And this slide shows you some, I would say, popular cleavable linkers that allow to release uncharged drug upon cleavage in the cell. That could be caused either by protease or by acidic pH. And it should be also mentioned that non-cleavable linker are also used, but overall, there is a majority of cleavable ADC linker used in the field. So as of today, the low therapeutic index of ADC remains really a great challenge to tackle for the industry. And a recent study estimated that at least 23 out of 55 halt or discontinued ADC was due to actually poor therapeutic index, actually preventing to reach sufficient efficient dose for in patients. So actually what has been happening during the last decade is that ADC toxicity is actually mainly off-target mediated, and off-toxicity is consequently still a major issue to solve. As you can see, following administration of ADC, it is distributed to the whole body. That means normal tissue, and then there is an enrichment at the tumor levels, which remains still limited. And the cellular uptake can take place both in normal and obviously in tumor cells, meaning that catabolism and release of the cytotoxic can occur in many organs besides the targeted tumor. And most of the time, toxicity, the so-called drug dose-limited toxicity of ADC is really due to target independent internalization of the ADC in normal tissue. And the linker, the combination linker and payload is really very important to actually limit this off-target toxicity. In the next slide, I will show you different strategies that are being developed to avoid this target-independent uptake in normal cells. So I will be very brief, but there is obviously a strategy to avoid actually the Fc gamma R mediated internalization by using, I would say, specific engineered mAb with reduced Fc gamma R binding. There is also various strategy to avoid the non-specific endocytosis by playing on the FISCAM properties of the payload and the antibody. There is also strategy to actually avoid deconjugation that could lead to a premature release of the drug, the cytotoxic drug in the circulation that could cause actually toxicity. And then the so-called bystander effect could be also limited by selecting payload with low membrane permeability, and also by using obviously non-cleavable linker. Once again, in that case, it depends about the indication, the target you are dealing with, because the bystander effect could be in some cases obviously beneficial. So in the next slide, I would like to maybe I can skip this one because it presents actually some, I would say, strategies to improve therapeutic index, but I prefer to go directly to examples. And this one actually is an example of technology advances allowing to increase the therapeutic index of ADC. This is actually the site-specific conjugation, and actually AstraZeneca researchers have reported nice results, as you can see, showing that the advantages of site-specific conjugation by using cysteine engineered mAb that allow to direct specifically actually the conjugation to the payload. And as you can see on the various graphs, when you compare with the conventional methods, that means the stochastic conjugation to lysine, there is really actually obtention of homogeneous ADC, a uniform composition, and it leads to actually an improved PK, an improved efficacy, an improved tolerability, and overall, obviously, an improved or an increased therapeutic index. So it's not always the same situation, but it shows you that the SSC could be really a breakthrough for the next generation ADC. But it's also fair to say that at this stage, the most advanced ADC are still using, conventional conjugation, and none actually ADC based on ECC has still reached actually a late stage of clinical phase. So another promising approach is based actually on what we could call local activation. That means activation at the tumor site specifically, and CytomX has developed a very elegant approach based on the Antibody-Drug Conjugates, where the paratop of the antibody that needs to will actually bind the antigen are protected with a masking peptide that will be released ideally and specifically in the micro environment of the tumor, where actually there is an overexpression of proteases that are able to cleave this peptide and allow the antibody to bind its target and then be internalized to kill eventually the cell. So this is really a nice approach, and you can see on the right hand side that there is already some favorable comparison between actually an ADC and a Antibody-Drug Conjugate, showing that this approach could be a valid one in clinical stage. So we need to have more data at this stage to make sure that it will go it will bring a new therapy for patients. The other actually emerging axis of development is actually concerning smaller scaffold to antibody. Since antibody are large, there is actually obviously limited tumor penetration, and as you can see, there is a lot of different formats, antibody fragment being actually explored. There is nothing well advanced, but as you can see, there is nevertheless two, even three molecules being at the clinical stage the bicycle peptide from Bicycle Therapeutics. We will see more data afterwards, and also two actually one peptide drug conjugates and one small molecule drug conjugate developed by Tarvida that are also actually small molecules and that are in early clinical phase. So one word about this bicyclic drug conjugates. This is actually using the technology developed by Bicycle Therapeutics. You can see that this BT 1718 is actually bearing this bicycle peptide that is able actually to bind an extra cellular target, actually a matrix metalloproteinase MT1. And this is a small, actually, molecule, since it's about 2 kD of molecular weight. And what is really striking is that it was able to outperform ADC in a head-to-head comparison that was actually presented at the EACR meeting last year. So this is something very promising. Obviously, tumor penetration will be increased, and we could also think about, again, of toxicity profile based on the fact that there is no FC on such, scaffold. The other one on the right-hand side is a peptide drug conjugate developed by Tarvida Pintarins that use actually somatostatin receptor to actually internalize payload, a methansine payload within the tumor cells. So at the end, I hope that I will convince you about actually the promise of the ADC field with so far 8 ADC approved and marketed. Obviously, there is still a need to actually find better drugs with increased safety, for sure. So there is a lot of effort being done in the pharma industry to find I would say the better match between the mechanism of action, the targets, the antigen targets, and also obviously the indication and the patient population. And obviously there is also a lot of work being done regarding the development of new formats besides antibody. There is some new release of bispecific ADC. There is also some kind of dual payload ADCs that could also maybe bring some benefits for the patient. And then there is also, I think, an interesting trend where we see ADC moving beyond oncology. We see advanced ADC for infectious disease in immunoinflammation, also inflammation area. So I think there is a lot of room for also positioning ADC modality in such field beyond oncology. I thank you for your attention, and I'm ready for questions if we have time. I don't know.
Thank you very much, Oliver.
Can I ask a very brief question? Very nice talk and very impressive results you have. With respect to the payloads, is there a size dependency on the payloads that you're able to actually put on the antibodies, or how do you go about that?
Well, I think it's possible to deal with various type of of payload. I think that what is maybe the most important is the charge and the hydrophobicity of your payload, because it does impact actually, for example, the off-toxicity profile. So there is many strategies going on for those I would say cytotoxic payload to try to decrease hydrophobicity, especially when you increase the drug antibody ratio. You have to face this issue. What we know, so it was not mentioned in my conclusion, but you're right, it's a good point. There is also a new type of formats. For example, antibody oligonucleotides conjugates are emerging. So obviously you have to deal with, I would say, molecules that are by far, bigger than cytotoxic payload, and also with a lot of charge. So it works. I mean, we can say it will work in human yet, but nevertheless, there is, at least at the preclinical stage, good indication that it's feasible. So there is really a broad range of payload that could be actually conjugated to antibody, but in each case with specific issue to solve.
Perfect. Thank you.
You're welcome.